We go from, this is our threshold, right here. And so then depolarization, that's the opening of the sodium gating, okay? Sodium gate's open. And we're gonna go up. How far do we go up here? This
To
was
positive
0.
Positive 20?
The illustration we were using was a plus 20 millivolts. We got up to that point, then what happened?
Then we start to go down the grind.
The third point was repolarization. I don't know, I think it's O-R-I-Z-A-T-I-O-N. Repolarization. Now, repolarization was the potassium gates are opening, right? And then we go beyond that point, we have this little hiccup in here before we get, I wanna make sure that that's about the same level here. So we have repolarization, hyperpolarization. And then we had to have what? What was the next step of this whole thing that had to take place?
The sodium potassium.
Sodium potassium pump. And then we got back to our MP. Okay, hyperpolarization. This is hyperpolarization right here. Did that make any sense? Huh? A little bit, yeah, okay. Resting membrane, minus 85, we were at minus 85 millivolts, okay? And then we get up to a plus 20, this was our area where we said it was a plus 20 millivolts. This is, usually they put this as a minus 50 millivolts. The threshold. Let's see if this makes any, I'm gonna take a few more minutes of your time for this video here.
A skeletal muscle contracts only when stimulated by nerve impulses from a motor neuron. Without innervation, muscles become paralyzed and eventually atrophied. The axon of a motor neuron usually gives out many branches, supplying multiple muscle fibers.
You see that? Dendrites, cell body, axon. That's exactly the drawing I have up on the board up here. It's just kind of a Xerox copy of it.
They're pretty identical.
Yeah, it's identical. I'm glad you appreciated that.
These fibers contract in unison when activated and constitute a motor unit. A motor unit can contain anywhere from just a few muscle fibers to thousands of them. Small motor units are found in muscles that require finer control. For example, muscles that are responsible for subtle movements of the eyes. Large motor units are found in larger muscles that require strength, such as muscles of the arms and legs. Muscle fibers of the same motor unit are usually not clustered together, but scattered throughout the muscle. This way, stimulation of a motor unit has a slight but uniform effect on the whole muscle, and not causing a small, localized twitch. The strength of a muscle contraction is determined by the number of motor units that are activated at one time. Even at rest, most muscles are in a partial contraction state called muscle tonus, which is maintained by alternating activation of a small number of motor units. The connection between a motor neuron and a muscle fiber is called a neuromuscular junction, which is basically a chemical synapse between the nerve terminal and a specialized area of muscle cell membrane called the motor end plate. When an action potential reaches the nerve terminal, it causes the release of the neurotransmitter acetylcholine into the synaptic space. Acetylcholine then binds to nicotinic receptors on the end plate. Nicotinic receptors are ligand-gated ion channels. Upon binding to acetylcholine, they open to allow sodium to enter the cells, depolarizing the cell membrane, producing the so-called end plate potential. An action potential is generated in the muscle cell only when the end plate potential reaches the threshold required to activate voltage-gated sodium channels located outside the end plate in the neighboring membrane. When activated, these channels allow faster influx of sodium, further depolarizing and eventually reversing the polarity of the cell membrane. At this point, voltage-gated potassium channels open for potassium to move out, quickly returning membrane voltage to its original rest.
Notice they have that plus 70 on that, do you see that? They didn't go to a plus 20 on this animation here. They start with a minus 95, they have a minus 50 as threshold. Minus 50 is fairly standard for the threshold, but here they're using minus 95, and then they go all the way up to a plus 70 here, which I've never seen one of these illustrations being used that. But I like their garbage can little reference to the gated, little gates going up. Now, if you're having some difficulty with what we've been going over this evening, I would encourage you to dig through a bunch of these. Some of them are gonna have a different take on it, just like this one. They're using, minus 95 and a plus 7. Get the concept. Don't worry about the minutia of the terminology, or excuse me, they say the nicotinic receptors right here. Okay, we're gonna have a little bit of a reference to that, but we don't go into great detail. But here we're talking about potassium channels and sodium channels. The sodium channels are opening out here. They're gonna be voltage gated here, and then we're gonna talk about, ligand gated, and these are gonna be voltage gated right here, and then we're gonna be moving the ions there in order to have the muscle contraction. Get the concept, okay? That's the big concern I have with this. There's so much information here, and if your eyelids were fluttering a little bit when we were doing this, you missed out on something. It's gonna be difficult as we move through this material here, because it just gets piled on to this concept right here, okay?
Value. Once generated, the action potential spreads,
I'm gonna back it up, 'cause I talked over a little of that.
quickly returning membrane voltage to its original resting value. Once generated, the action potential spreads like a wave, thanks to similar voltage-gated ion channels located throughout the muscle fiber. The action potential also runs deep into the fiber via T-tubules to reach the sarcoplasmic reticulum. Here it activates voltage-gated calcium channels, releasing calcium from the sarcoplasmic reticulum into the cytosol of muscle cells. Calcium then sets off muscle contraction by the sliding filament mechanism. This mechanism is described in another video. Another important component of the neuromuscular junction is the enzyme acetylcholinesterase. This enzyme removes all acetylcholine molecules that do not immediately bind with the receptor, and those that are done activating a receptor. The enzyme action essentially terminates synaptic activation, giving the muscle time to relax, and thus preventing continuous contraction that would result in muscle spasms. Substances that cause muscle weakness or paralysis do so by interfering with the function of neuromuscular junction. Botulinum toxin prevents acetylcholine release from the presynaptic side of the junction. Some other toxins attach to nicotinic receptors, blocking acetylcholine from binding but do not open the ion channel. Certain drugs lodge into the channel of nicotinic receptor, blocking the passage of sodium. All these substances prevent activation of muscle cells and cause flaccid paralysis.
Okay, what they're showing you here is that right at the head here, it says neuromuscular toxins. Okay, and one of the things that I mentioned, I think I just mentioned casually or previously, is that when we talk about Abbott and Baxter and Pfizer and whoever other chemical pharmaceutical companies out there, they spend a whole lot of research, time and effort into figuring out how we do some alteration of this 1/1 millionth gap space here. because we can have, as it mentioned here, the botulinum toxin or Botox, which is used, the medicinal form of that, which can be used for muscle spasms, cramps and stuff like that. We can put something that in there as an agent that can interfere with the neuromuscular junction. We go over here in curare, the plant toxin, which is, and another one they have here is a snake venom, but we hear about in South America where they had the blow guns, the blow darts, and they're using curare. But what it does is it's a, it is a plant toxin, but what it does is it blocks the receptor sites, okay? Remember ligand gated? You need to have the right card to open up that gate. Well, if you block that binding site with a neurotoxin, you then can paralyze that individual, and that's what the blow darts do, is that they give a, it's suggested that if you have somebody that can give you respiratory assist for about 10 minutes or so, you can survive that because it causes a paralysis of the respiratory muscles, okay? But the problem here is that without that, those people use those blow darts to harvest animals, to get their food. Because you hit an animal with enough of those, it paralyzes them, and then they can go and actually kill it. Then we have one over here, and I don't remember what was underneath this information right here. But there's a number of toxins which we have that are capable of interfering with those points of the neuromuscular junction. Listen to those again. They're worth hearing, I guess.
Botulinum toxin prevents acetylcholine release from the presynaptic side of the junction. Some other toxins attach to nicotinic receptors, blocking acetylcholine from binding but do not open the ion channel. Certain drugs lodge into the channel of nicotinic receptor, blocking the passage of sodium. All these substances prevent activation of muscle cells and cause flaccid paralysis. On the other hand, some pesticides inhibit acetylcholinesterase, preventing degradation of acetylcholine, causing continuous activation of muscles. That's how they induce muscle spasms and cause spastic paralysis.
All right. So there's a whole lot of this information that's available out there online for further study if you're so inclined, but the key thing about it is to try and help you feel some confidence in what we're talking about right now with this information. All right, let's do a little bit more on this. Stop the screen here. All right, so here's muscle twitch was one of the, they were just mentioning about flaccid paralysis and spastic paralysis and some things such as that make a little sense. We do talk about when we're dealing with a stimulus that's applied and there's a lag, a contraction phase and a relaxation. So it's the muscle twitch physiology, the responsive muscle to a single action potential along a motor neuron. We have a lag or latent, we have a contraction phase here, and we have a relaxation. Well, in order for the contraction, we have to have the calcium in there, okay? We have to have the beginning of it, we have the calcium release, and then the calcium returns here, so it has to be a relaxation phase here. You might hear about some things called isometric and isotonic contractions. Isometric exercises were a big deal, you know, about 40 years or so ago. We hear about them from time to time, but no change in length, but tension increases. It's like you know, we're gonna say, why don't you come over here and help me move this wall? Okay, and so we get all of you leaning against the wall. Push harder you can, push harder. That's an isometric type thing, and then you relax, okay? You do isometric exercises. What does it mean? No change in the length, but the tension is gonna increase. You're pushing against it, and you're just gonna continue to push like that, okay? That's isometric, okay? Isotonic, change in length, but the tension is constant. You know, change in length what we're doing when we're talking about doing some weight training types of things like that. So the motor unit. That was mentioned in the last video where we talked about the motor unit type of thing here. A single motor unit motor neuron, and all the muscle fibers innervates, okay? This neuromuscular junction thing that I have right here, okay, the motor neuron and all of the muscle fibers innervated by it. An action potential in the neuron of a motor unit causes all the muscle fibers of that particular area to contract. Okay, and so here we have not just one, but we have all of these coming from this one axon of the motor neuron. I'm giving you one individual little piece of the end plate where it makes contact, but in this case right here, they're showing you how that's more likely to be the case where you have all of these. The motor unit, larger muscles have motor units with many muscle fibers, okay? Like they showed you, the gastrocnemius muscle, the posterior aspect leg, or we're talking about the smaller muscles in the eye. The make up delicate movements contain motor units with few muscle fibers, okay? We had rectus muscles for eye movement. We talked about the 6 muscles that are associated with what are called extrinsic eye muscle. All right, the strength of a muscle contraction varies from weak to strong. The muscle responds in a graded fashion depending on the force generated in the individual muscle fiber. Have any of you ever picked up a pencil? Ever picked up a 2-year-old? Okay, you ever pick up a glass of water? You pick up a good-sized stone? Do you use the same amount of force with every one of those? How do you make the difference? How do you make the distinction between, I'm gonna pick up this mug right here, or I'm gonna pick up a 2-year-old? If any of you ever picked up a child where you thought, well, this could be heavy, and this little feather light, or you see this child and you think, and it's really glued to the floor. Wait a minute, 40-pound 1-year-old, here, what's happening? I mean, I've done this with a grandniece or something like that. I was so cute, look at this, is a little tank. Do you know what I'm talking about? All right, you pick up a styrofoam cup versus a glass. You understand what I'm talking about? Do you use the same amount of force? Why not? I mean, you're not likely to crush the glass, but you will the styrofoam cup. Do you follow what I'm talking about? And sometimes we get fooled by it because, and even landscape people sometimes have these, what are they, styrofoam rocks that make, look like a rock there, and then it's supposed to be the illusion of a stone to conceal something. Well, looks like a stone to me. The muscle responds in a graded fashion depending on the force generated in the individual muscles. To a certain extent, that's prior knowledge information, and I have been fooled by that on a number of occasions. Something looks like it's going to be real heavy and it's not. Something looks like it's real light and it's really heavy. Okay, increasing number of cross bridges allows a fiber to contract with more force. We do that, kind of that's learned behavior too. Factors that increase the number of cross bridges are the frequency of the stimulation, the muscle fiber diameter, and muscle fiber length at the time of the contraction. Is it stretched out or is it already partly contracted when you're beginning this? I'm not going to deal with treppe. You got that in your notes, dump out the treppe. We're not going to do it. You're going to hear about, from time to time, things called tetanus. Some of you have had, I would guess that all of you, if you've got a shot record at home, you've had a tetanus shot, right? You probably have it as, diphtheria, pertussis, and tetanus. You had a DPT inoculation. Diphtheria, corynebacteria diphtheria, Clostridium, we have the, excuse me, let me try again here. We have the diphtheria, we have the pertussis, which is the type which we usually associate with something called whooping cough, and then we have the tetanus, which is Clostridium tetani. Now, why do you have those three? Well, they're considered kind of important, inoculations for us. Well, tetanus is one of those where we have tetanic spasms. Something goes into the lock up here. And we usually talk in terms of having a tetanus shot when we make reference to a person that you've just stepped on a rusty nail, or you have some kind of anaerobic type of microorganism. Those of you in the microbiology might appreciate Clostridium botulinum, Clostridium tetani, Clostridium perfringens. Those are all anaerobic microorganisms that live in the soil, and they get into our body here. They don't need any oxygen at all, but they're neurotoxins. Remember that illustration we just saw? A neurotoxin, what is it doing? It's stopping something from happening that's supposed to take place here. And we talk about tetanus here as being a locking up. But when we hear about lockjaw, the origin of lockjaw was from the cavalry soldiers who were always around a whole lot of horse manure. Well, horse manure contains a lot of Clostridium tetani. You got a puncture wound, you got a saber cut, you got some kind of a gunshot, and it seals up a little bit, and you end up with the Clostridium tetani microorganism in you. It starts producing neurotoxins. And what happened? Those soldiers that they studied here were doing that. They just lock up like that. And they said, oh, they obviously died because they couldn't eat. They're lockjaw. Well, what's one of the other major muscles that allows you to survive? What are you going to say?
Your heart.
The heart? Good choice. Anybody want to weigh in? Something else? No, that's not a muscle up there. Don't point to that. That's not the muscle you're looking for. How about the diaphragm? What's the diaphragm all about? Respiration. These guys didn't die from not being able to eat. They died because they couldn't breathe. You locked up all the muscles of respiration. And so the Clostridium tetani, but then Botox was mentioned over here. Remember what locks up the sites here? What is a Botox? Botox, botulinum toxin, is one of those that lives, again, anaerobic microorganisms, but now we figured out we can use a little bit of it. You don't like those little claw feet around the edge of your eye? Let's put a little Botox in there. You got back spasm? Let's put a little Botox in there. Medicinal Botox. Not industrial strength, medicinal version of it, okay? It's a neurotoxin. Can it kill you? Yeah, if it's not used appropriately. It's a neurotoxin. What is it trying to do? We're isolating it to our benefit for medical purposes. Okay, so tetanus is a real deal. No relaxation between contractions. Incomplete. Muscle fibers are partially relaxed. Wave summation, another thing that calcium ions cannot be removed fast enough when the muscle fiber is unable to relax in a tetanic spasm. We talk about tetanus. I'm not going to do that. All right, we do talk about muscle fiber diameter, okay? The greater the muscle fiber diameter, the greater the force of the muscle fiber can generate. Does that make sense? Doesn't that sound like, yeah, I guess I can buy that, right? You got a bigger muscle. You got a bigger rope, you can put more force on it before the rope's going to snap. Take a thread and you pull on it, you're going to break it. You take a 1-inch rope and you pull on it, and you're not going to break it, okay? Kind of what we're dealing with here. In general, larger diameter fibers have more myofibrils. That should make some logical sense. Larger diameter, more myofibrils. Therefore can form more, excuse me, more cross bridges. Yeah, are we clear with what a cross bridge is? I'm not getting an amen on that, I'm not sure. What was a cross bridge?
That link between.
Really? Come on, wait a minute. Really? Are we clear on what a cross bridge is? Help me, come on, elaborate. The binding of what?
Myosin heads binding to actin.
Good answer. That's a cross bridge. Yeah, that's the right answer. We're looking for a myosin head linking up with an actin binding site. In general, larger diameter fibers have more myofibrils. If they have more myofibrils, more of these sarcomeres in there, we can form more cross bridges, more force of contraction. That should make some sense. Are we good with where cross bridges are? Okay. Muscle length. Active tension. Force applied to an object can be lifted when a muscle contracts. Does that make sense? A muscle contracts means what? The two ends come together, correct? Muscles have to be attached to a bone across a joint. Makes no sense to have a muscle attached to the bone right here and to the same bone down here. For what purpose? You cross a joint with a muscle in order for the muscle to shorten, and when the muscle shortens, it activates that joint surface there. Active tension increases or decreases the muscle fiber changes in length, the active tension curve they talk about. Stretch too far, fewer cross pieces. If you really are stretched, have a muscle already stretched out, and then you try and do some action with it or perform a contraction, that's going to be difficult because you already have this muscle at a stretched point here, and it's not going to be able to fully activate all those cross pieces. If not stretched at all, thick filaments touch the Z discs, and very little contraction can occur. If we already have the muscle in a state of where the Z discs are, remember the Z discs? I told you remember Z discs. And I said remember actin and myosin. What attaches to the Z discs? Is it the myosin or the actin?
Acting.
The actin through the titin, okay? Passive tension, tension applied to a load when muscle is stretched, but it's not stimulated. It's just some passive amount of tension. You have some tension on a muscle here. Yeah, some of the active. Here's muscle length 1. Is there gonna be much capability of performing any action here? Can you see this one? Here's the Z discs. Whereas the actin is almost touching one myofilament to the other, the contractile protein actin is just about touching. Are we gonna get much of a contraction on that? Probably not. What about here? Yeah, here we've got maximum capability for it because we have, but then at this particular point, we're way out at the end point here, it's gonna be really difficult for this stretched muscle to be able to perform some work or action as a consequence of where that muscle is. Recruitment. Let's see, what do we have? Subthreshold. Okay, recruitment. What's a subthreshold stimulus? Okay, what was threshold first of all? Give me a value, a number value.
-50.
Minus 50 millivolts, right? So what's subthreshold?
Below.
Yeah, something that is more negative than minus 50. Subthreshold. Can you have an action potential if you're subthreshold stimulus? Why not? You're doing this and saying no. Why not? Yes!
All or nothing. All or
But you had to reach what first?
threshold.
You had to reach threshold. Yes, exactly. That was a really good answer. You have to reach threshold first, and then it's all or nothing. If it's sub-threshold, did you reach threshold? No! There can't be an action potential if you're sub-threshold. If you're getting down here and you're only getting up to this point right here, well, you're not quite the threshold. You gotta get over the threshold. Okay, so sub-threshold, no action potential. Can't have a contraction. That should be duh. Okay? Threshold stimulus, what does that tell you? Action potential, there's gonna be something going on. It's all or nothing. Maximal stimuli, that's a little confusing. It says stronger stimuli than that produces action potentials in axons. Now we're jumping ship here. We're going to additional motor units get integrated in this. And maximal stimulus action potentials are produced in axons of all the motor units of a muscle. A greater stimulus, a supramaximal, has no additional effect. Go ahead, keep stimulating. It's not going to do any more. You're already maxed out here. The size principle. During recruitment, small motor units are recruited first. Well, yeah, you don't give the big guns right away. You're going to start off with, let's test the water here. What kind of muscle or what kind of action potential do we need here? Muscle tone. This is the one we hear a lot about. We talk about people that are, we make reference to the under-exercised, underutilized. We talk about the absence of muscle tone, of good muscle tone here. It's a constant tension by muscle for long periods of time due to small percentage of all motor units contracting out of phase with one another. That gives us tone, the muscle tone. The idea that there is good muscle tone there. This person is capable of doing some activity. This person has the muscle integrity there to be able to do something. Without the muscle tone there, we've got some problems here. We see that in a person that's inactive. You know, we talk about those that are bedridden, so to speak, or they're individuals such as that. Isotonic, change in the length, but the tension is constant. Just doing an activity like you're curling a weight or something like that. Concentric, overcomes opposing resistance and the muscle shortens. Overcomes a concentric here. And then eccentric is the tension is maintained, but the opposing resistance is great enough to cause the muscle to lengthen. You're just kind of out at the extreme here. All right, we're gonna get that. I have 10 questions for you guys. Is that right? Are we at 9:20? Yeah, we're close. Close enough. I have 10 questions.